Hello, everyone. My name is Carla, and it's an honor to be here today to present part of my PhD work at Robert Pasillo's lab, and which consists on developing of a novel micro-engineered hard tissue platform on chip with multicellular biomimicry. So first of all, as you may know, cardiovascular disease is the first cause of mortality worldwide, and currently there are not really treatments to cure this disease. And actually 6% of clinical trials are currently ongoing to try to find new treatments for this disease. But unfortunately, there is a low success in the drug discovery. And additionally, a lot of other drugs also exhibit some sort of cardiac toxicity. So that's why actually the human pluripotent stem cell-based models are very useful to incorporate in the new drug discovery pipelines to be able to stratify more the population and try to find more treatment in a more personalized way. But for that, then, we are looking at cardiac models, right? And as you may know, human pluripotent stem cell-derived cardiomyocytes are actually very immature and don't exhibit the complexity of the adult human heart. Even though we can actually extract a lot of readouts already from single-cell experiments and 2D, 3D models are most commonly used to try to reach this complexity of the adult human heart. However, we are still facing some limitations, and one of them is the circulation of the blood towards the cardiac tissue. So for that, we actually thought of developing a heart-on-chip platform where we can actually culture EHTs, so engineered heart tissues, in each of these individual chambers that you see here, and they are also interconnected, so the flow of media can actually go through each of these chambers. In each of these chambers, we actually have two PDMS posts. And then when we put the cardiomyocytes together with the cells of interest, we can actually measure the force of contraction of these tissues by measuring this displacement of the tissue. And this is actually made with only 50,000 cells. And actually, because we are putting a microfluidic system in here, we wanted to see if this already has an effect on the functionality of the tissue. And we saw that indeed, when we culture the tissues in a rocker or in a pump system, actually the force of contraction increases. But of course, the cardiomyocytes in the heart are not directly exposed to the fluid flow, right? So they are shielded by a layer of endothelial cells, as we just heard. And actually, this interaction with the endothelial cells is also known to improve the cardiac maturation of the cardiomyocytes. So for that, we also wanted to have this as a main goal of our work. And we wanted, first of all, to keep this microfluidic system to be able to perfuse. We also wanted to maintain this EHT format to be able to extract one of the main functional readouts of the cardiomyocytes, the force of contraction. But also, we wanted to introduce new cell types to try to mimic this in vivo-like cell organization. So for that, we started first with mixing the cardiomyocytes and the fibroblasts that we always actually use as a culture system, and then we introduced fibroblasts and endothelial cells and smooth muscle cells. Actually, this is a bit of a long talk if I would have to explain a lot how I optimize all these ratios, but I tried to summarize it here basically by explaining that we had to really fine-tune the amount of cardiomyocytes that we were having in the system to be able to preserve their structure and organization and their performance assessed by force of contraction, but also to recapitulate the presence of the other non-cardiomyocyte subtypes. And then the results that I will show you in the next slides are actually compared with this control system, which we call CF, which only contains the cardiomyocytes and the fibroblasts. So the first observation that we had is confirming also previous studies that this endothelial cell addition actually improves the force of contraction, the contraction properties of the micro-EHTs. We saw that by an increase in the contraction force, but also with an increase in the post-rest potentiation, which measures the sarcoplasmatic reticulum of the cardiomyocytes, which is a better sign of handling of the calcium. We also saw an increase an increase in the frequency pacing. So when we paced the tissues to high frequencies, we saw that they were able to follow this in a more synchronized way. And as you see here in the controls, actually you see that at 5 Hz, there were already some aberrations with the control tissues. We also observed in electrophysiological tests that the action potential duration at 90%, so the repolarization of these tissues, of the cardiomyocytes, also increased, but also importantly, the conduction velocity also increased significantly, which is also a sign of a more mature tissue. We also confirmed with calcium dynamics, and although we didn't see differences in time to peak, we did observe that the decay time was reduced in these tissues. We further confirmed this interaction between the cardiomyocytes and the endothelial cells with RNA sequencing data. And first of all, these gene ontology terms already point out that we also saw an upregulation of calcium ion homeostasis genes, and also confirming especially that the transcriptomically, the endothelial cells were also active by, for example, expressing genes related to vasculogenesis or the endothelial cell proliferation and development, which that was confirming that they were active. But we also check for some genes that are known to be involved in the crosstalk between the cardiomyocytes and the endothelial cells. And we actually also found that, for example, apelin, the signaling factor, and both the receptor were expressed in our data set, also confirming this interaction between the cardiomyocytes and the endothelial cells. And going back to the point of the higher conduction velocity that we observed functionally, we also observed a higher expression of connexins, which are also known to conduct better the signal through the cardiomyocytes and the endothelial cells. Then we also saw morphologically how are these cells interconnecting. First of all, we saw that the endothelial cells here in yellow were found also interconnecting with the cardiomyocytes. We also observe, interestingly, that the endothelial cells in yellow were actually like all interconnected with the fibroblasts that you see here in red. So they were like forming like a, kind of an interconnected network altogether. But also, most importantly, that by checking connexin 43 expression, the cardiomyocytes were also interacting with the fibroblasts and the endothelial cells. But then, as I mentioned at the beginning, when we actually make this ratio optimally, we observe this conformation in a very success rate of formation, where the endothelial cells are actually wrapping the tissue. So they are in the inside of the tissue, but actually mainly they tend to form this wrapping layer around the whole tissue, as you see here in yellow. So when we tried to quantify this cover layer, we actually saw that almost in 80% of the cases, the endothelial cells were forming this layer. But we thought, okay, why is this happening, right? And then we checked first, are the smooth muscle cells playing a role in this layer formation? So to test that, we removed the smooth muscle cells of the culture, as you see in this group here. And actually, we confirmed that the smooth muscle cells support this endothelial cell layer formation. Here you have an example of how the tissues look just in the Vicatherian staining when the smooth muscle cells are removed. And if we quantify that, we also see that the endothelial cell coverage decreases. And then we thought that this layer perhaps was playing a role in as a functional barrier. So we tried to quantify that with a dextran assay. And in this case, we compared, of course, back to our control, the cardiomyocyte and fibroblast only, and then we diffuse the tissue with dextran. And then we tried to see how was this, the permeability of the dextran through the tissue. So you see that in here, in red, are the endothelial cell layers. And then, when we quantify that, basically we see that the permeability of the endothelial cells, that they are acting as a barrier by showing a reduced permeability. So we try to exploit this property by checking how different drugs would affect in this system with this layer of endothelial cells, since in vivo the drugs need to pass through the endothelial cells to get to the cardiomyocytes. And actually we saw that drug responses are delayed in this system, as you see here, by a increase in force when we treat with an inotropic agent, like a positive inotropic agent like isoprotenolol. We also see a delay in the reduction of the force with a negative inotropic agent like verapamil. And also when we treat the tissues for 2 days with doxorubicin, a cardiotoxic compound, we also see that actually this reduction of force is also decreased up to 24 hours. So then, in a next step, we thought that since these endothelial cells in vivo also play a role in fatty acid delivery to the cardiomyocytes by transporting actually the fatty acids from the blood with specialized transporters to the cardiomyocytes, and this actually affects their functionality as well. We checked first in our RNA sequencing data how was the expression of these known proteins, well, in this case genes, that are involved in this network. So these are mainly the ones that I point out here, so C36, LPL, LIPG, and also GPI-HBP1. And actually we saw that there was a trend to increase actually, and some of them were significantly upregulated. So to further look into this, we decided to use a fatty acid media to test further if this would enhance, potentiate this effect. And for that, we have here all the 4 conditions that I use. So we have the CF in cardiomyocyte media. It's a very specialized cardiomyocyte media. We call it control. And then also treated with a fatty acid media that we also developed. And then we also have the group with the endothelial cells and both the other 2 medias, of course. So when we do that, this is very preliminary data, but I'm quite certain that we are already seeing an increase also when we culture these tissues in the fatty acid media with the endothelial cells by seeing a trend to an increased expression of these markers, CD36, LPL, LIPG, and then especially after this is delivered to the cell, so you would expect the fatty acid going to the cardiomyocyte has to process this. And usually the CPT1 beta is a very common marker also to see the mitochondrial activity and just uptake of fatty acids into the mitochondria. And we also see that these tend to increase in this condition. Therefore, I would summarize what I showed you today, which is the development of a heart-on-chip model with heterotypic cell composition, preserving the EHT conformation for functional readouts. I also showed you that the addition of endothelial cells and smooth muscle cells improves the cardiac performance and the electrophysiology of the tissues. And finally, that this self-organized endothelial barrier influences drug dynamic and fatty acid transport. And this brings up new opportunities for drug screening and for study metabolic diseases. And last but not least, I would like to thank all the members of the team that actually helped a lot during the process of this project. And I'll be happy to answer your questions.
Now, floor is open for question. Do you have any in the middle?
Thank you very much. The ratio of the different cell types, is this based on the native composition, or did you do some work in optimizing?
Sorry, I cannot hear you much.
The ratio of the different cell types, is this based on the native composition, or was there some work in optimization?
Yeah, so the way that I started was basically checking a very previous report using pluripotent stem cells and trying to model the composition of the human heart as well in vitro. So that is how I started trying what composition was the best one, but I also wanted to go more towards the in vivo-like composition. So I tried different ratios in this case, and actually I have here this slide as well because I thought that could come. So maybe the one that is mostly described is 70% of cardiomyocytes, and then you have a mix of endothelial cells and fibroblasts, 15-15, that is very commonly established. But I noticed that in my case, as I mentioned, the representation of the endothelial cells was not very strong, so it was there, but accidentally sometimes I saw that if the ratio was different, then I was starting to have this layer formation. So then I tried to go a bit back in this optimization and start from the very beginning with a very low percentage of cardiomyocytes, and then increase from there towards what we know. And I set up the threshold as I mentioned, with a good organization of cardiomyocytes, as you see here, and also exhibiting a good force of contraction, and also having this representation of the endothelial cells. Yeah.
Thank you. Question at the back.
Very interesting talk. I actually have two questions. First of one is, I was wondering if your extracellular matrix model could associate the inotropic effects that you observed to explicitly to each components of non-cardiomyocytes, like collagen, for example, if you have considered them. And my second question is about the maturation, because you said that those cells were in maturation process for 10 days, if I'm not mistaken. I was wondering if you have considered other intervals, or if you would say that, for example, 30 days of maturation, because this is a protocol that I routinely actually see. So if, for example, longer intervals of maturation would affect your results. Thank you.
Yeah. So going back to the ECM, I saw that a lot of ECM genes were upregulated with the addition of the endothelial cells. And while they pop up here also with the gene ontology terms, I think if I go a bit back, you can also check it. So I believe, exactly, ECM organization basically. So I saw a lot of genes related with collagens, and laminins as well were also upregulated. So yes, in this sense, there's a lot of ECM remodeling as well. And going to the second question regarding the days of culture, so usually we perform the tissues, we make the tissues when the cardiomyocytes are at day 20 already, and then we continue from there for extra 10 days of culture. So they are already at day 30. But then, of course, you can, for some experiments, you have to go longer in time. Like I kept them even 20 days after making them, and they are still beating properly, and the endothelial cells, importantly, are still there. But in terms of force of contraction, I think that day 10 is actually the best timing to already have your readouts. So, yeah, I hope that answers.
Thanks, Carla.
Yeah.